In a study conducted by Oberlander et al., researchers investigated the effects of maternal depression on the gene NR3C1, an infant cortisol stress response. Ultimately, the question researchers wanted to answer was whether the prenatal exposure to maternal depression influenced the stress response of a child by altering the NR3C1 gene. Additionally, researchers sought to find out whether the use of antidepressants resulted in differences in this gene. While postnatal exposure to maternal depression affects the development of the limbic system, which is responsible for emotional response and regulation, researchers wanted to see if prenatal exposure affected genetic expression and the structure of the brain. Based on this information, researchers predicted that prenatal exposure to depression would increase infant cortisol response. 82 mothers were broken up into 3 groups: those who were depressed and not treated with antidepressants, and those who were depressed and treated with antidepressants. Non-depressed mothers served as a control for depressed mothers, whereas untreated mothers served as a control for treated mothers. Maternal mood inventories were taken at the start of the study, the 3rd trimester, and 3 months postpartum in order to measure levels of depression. Additionally, DNA was analyzed for the expression of the gene NR3C1 by obtaining blood samples from the mothers. At 3 months, infants were tested for a cortisol response. The cortisol content of infants was measured by taking 2 saliva samples in response to a stressor. One sample was taken as a baseline before the stressor was introduced. The second sample was taken post-stressor as the recovery level. The 2 were then used to calculate a cortisol change score. Additionally, neonatal cord samples were also taken to measure the expression of the NR3C1 gene in infants and compared to those found in their mothers. After the testing, researchers found several associations between the measured variables. First, increasing maternal depression scores were associated with the increased methylation of the NR3C1 gene both in mothers and their infants. Furthermore, variations in the methylation of the NR3C1 gene were associated with changes in the cortisol change score in infants at the alpha equals 0.01 significance level. Finally, there was no association found between the methylation of the NR3C1 gene and the exposure to antidepressants. These results were well aligned with what the researchers predicted, indicating that increased maternal depression was associated with an increased stress response in the infants via an epigenetic mechanism. Maternal mood was a key predictor of the changes in the NR3C1 gene associated with increased stress response. Because there was both a correlation between changes in this gene and stress response and changes in mood and gene expression, researchers concluded that prenatal exposure to maternal depression has an influence on emotional. On the other hand, the investigation of the effects of antidepressants is not as clear in its results. Antidepressant use has been associated with neonatal withdrawal symptoms and has been shown in animals to affect brain structure.